Hello guys, this is Muhammad and this is my first lecture with you. Basically, we are starting a series of lectures about the insulin in the physiology and pharmacology and then the diabetes mellitus. And today's topic is about the insulin synthesis. Before going into the details of the insulin synthesis, I want to talk about the pancreas, especially the endocrine pancreas, briefly. You know the pancreas is a long elongated organ, lies posterior to the greater curvature of the stomach, which is about 12 to 15 centimeters long and only 2.5 centimeters thick. In the pancreas, we have both exocrine tissue and also endocrine tissue. The exocrine tissue, which are clusters of exocrine cells, produce digestive enzymes directly into the pancreatic duct and then into the duodenum. Right? These are called the acini, and this is the acini cells, exocrine cells, produce the pancreatic juice, which contains many enzymes and alkaline juice, and secreted into the pancreatic duct and then into the duodenum, and help digestion of foods, carbohydrates, lipids, proteins, any other food particles. In the pancreas, we have also endocrine part, which is called islets, islet of Langerhans. These islets are small clusters of ovoid or spherical masses that are scattered in the pancreas and mainly located in the tail region. This part, is called the tail region. Here is the tail region, central body, the neck, and also the head. The islet of Langerhans are mainly located in the tail region. Humans, which have about 1 to 3 million islet of Langerhans, and each islet contains few hundred cells of endocrine cells and has a diameter of about 0.1 to 0.3 millimeter. Okay, about the cells of the islet of Langerhans, first we study alpha cells, or sometimes called A cells. Alpha cells, the cells that produce and secrete the glucagon, a peptide hormone. And also GLP-1, glucagon-like peptide 1. GLP-1 is important in the secretion of the insulin, while the glucagon, when the blood glucose level is low, for example, between the meals, in the fasting state, in starvation overnight, the alpha cells are stimulated and increase the blood level of glucagon. Glucagon directly goes into the liver to increase glycogenolysis, means breakdown of the glycogen into glucose. The preformed stored glucose form is what? Is glycogen. Glycogen is converted into glucose in the presence of glucagon, and also increase gluconeogenesis, means production of glucose from non-carbohydrate sources. Glucagon also goes into the adipose tissue to increase lipolysis. Okay, means conversion of triglycerides into fatty acids, and this fatty acids is then used by the body cells, especially the skeletal muscle and also the heart, for providing energy and ATP. The second type of cell that we have in the islet of Langerhans is called what? Beta cell, or sometimes called B cells. B cells secrete what? Insulin. Opposite to the state of glucagon, when the blood glucose level is high, when there is an abundance of energy after a meal, especially, the blood glucose level, when it's high, it stimulates the insulin. And insulin, in turn, helps for the proper utilization and also storage of the glucose. For example, excess glucose can be stored in the form of glycogen. Excess fatty acids may be stored in the form of triglycerides. And even insulin increase the uptake of what? Of amino acids. Insulin increase the uptake of the amino acids by almost all the cells in the body, and this amino acids inside the cells is used for the formation of new proteins. And insulin inhibit the breakdown of the of currently preformed proteins. Inhibit what? Breakdown of proteins. Right? But remember, the insulin is not the only product from the beta cells. Beta cells also produce C-peptide. This is the C-peptide. Also proinsulin. Proinsulin is an intermediate product inside the beta cell that's then cleaved, break down by the enzymes into C-peptide and also insulin. Proinsulin is break down into C-peptide and insulin. Also, the beta cells produce a very small amount of another peptide hormone, which is called what? Guess it. Amylin. Amylin is a peptide hormone important for the GI tract function and also increased satiety, feeling of fullness. Okay? The alpha cells and the beta cells are the most important cells in the islet of Langerhans, but also other cells are also present. For example, the delta cells. Delta cells produce somatostatin. Somatostatin is known as an inhibitory peptide hormone. It inhibits the secretion of insulin from the beta cells. Somatostatin inhibits the secretion of glucagon from the alpha cells. Also in other parts of the body, for example in the hypothalamus, somatostatin inhibits or suppress the secretion of growth hormone, inhibit the secretion of thyroid stimulating hormone. That's why somatostatin is called a universal or general peptide hormone inhibitor, because it inhibits the secretion of many peptide hormones. Other types of cells include the F cells. F cells that secrete the pancreatic polypeptide. These cells also called PP cells because they produce pancreatic polypeptide, PP, or previously called gamma cells, and also epsilon cells. Epsilon cells produce a peptide hormone which is called what? Ghrelin. Ghrelin and pancreatic polypeptide are important for regulation of the GI tract function, motility, and also regulate the appetite. Okay? If we enlarge one of the islets of Langerhans under the microscope, we can see how these cells are arranged inside the islets. Let's suppose this is a islet of Langerhans. Okay. In the islet of Langerhans, the beta cells are located in the center of the islet. And constitute about 60 to 70% of the islet volume. The most abundant cells are the beta cells and located in the center of the islets. The alpha cells are around the beta cells, and they are about 15 to 25% of the cells. Other cells include the delta cells, F cells, and also epsilon cells are less in number, and they are scattered among the alpha cells and the beta cells. Okay. These cells, of course, should be supplied with blood, and an arteriole directly goes into the beta cells. This is important because the beta cells are the most important cells to sense the blood glucose level and adjust the insulin secretion accordingly. And then the capillaries goes into the periphery to supply the alpha cells, delta cells, F cells, and also epsilon cells. The full completion of the beta cell mass is established within the first 5 years of life. Thereafter, the proliferation of the beta cells is very low, with an extending half-life of about, or lifetime of about 25 years. The beta cells have a lifetime of what? 25 years. But in the periphery, away from the alpha cells and beta cells, there are some immature cells, or called stem cells. These stem cells can be differentiated into what? Into beta cell when needed. For example, in the cases of damaged beta cells, or when the beta cell is lost. In the case, for example, pathological, in the case of diabetes mellitus type 1, beta cells are destroyed by the immune system. In the early course in the disease, These immature cells can be differentiated to replace the damaged and lost beta cells. Even the alpha cells and the delta cells have the ability to transdifferentiate into the beta cells in some cases, but ultimately the pancreas will fail to produce and secrete sufficient amount of insulin. Around the islet of Langerhans, there is a very thin reticular connective tissue capsule. This capsule is here around the islet of Langerhans. Is made of what? Connective tissue. And separate the islet of Langerhans from what? From the acini, exocrine tissue. These islets are surrounded by exocrine tissues. Here are many acini. These clusters of exocrine cells called what? Acini. And so on. Okay? That's why they are called islets, because the endocrine cells are separated by exocrine tissue. Here is an islet, then exocrine tissue, and then another islet, then another exocrine tissue, and so on. And also named Langerhans because first it was discovered by a German pathologist named Paul Langerhans. That's all about the islet of Langerhans. And now let's start to see how the insulin is synthesized inside the beta cells. Okay guys, let's start with the insulin synthesis, and suppose this is a beta cells, and of course inside the beta cells there's a nucleus. In the nucleus and in chromosome number 11, in the chromosome number 11 we have a gene which encodes the insulin. This gene is here that encodes for insulin. First of all, this gene should be transcribed into messenger RNA. This is the messenger RNA for the insulin, and this messenger RNA will diffuse out from the nucleus through the nuclear pores. There are many pores in the nucleus, and this messenger RNA, mRNA, is diffused out, and here we have what in the cytosol? We have, of course, ribosomes. Ribosomes are waiting for the messenger RNAs. The ribosome starts translation of the messenger RNA and make a chain of amino acids, a peptide. Okay, first amino acid which is added is what? Of course, it's the N-terminus of the methionine, and then other amino acids are added to the chain. Because the N-terminus of the methionine and other amino acids that are beginners in the translation of the messenger RNA are too hydrophobic, or we can say it's lipophilic, it recruits the messenger RNA, the ribosome, all this structure into what? Into the membrane of the endoplasmic reticulum. Here we have what? We have, or let me draw it in another color. Here we have the endoplasmic reticulum. This, we say this is the first amino acid which is too hydrophobic or lipophilic, directly enters the endoplasmic reticulum, and the ribosome is attached to the endoplasmic reticulum. So this endoplasmic reticulum is called what? It's called rough endoplasmic reticulum because the ribosomes are attached to it. And here is a messenger RNA. Right? The first ribosome is made a few amino acids joined to each other and make it the first part of the peptide. The other ribosome adding more amino acids, and the other one more and more and more until chain of amino acids or a polypeptide is formed. This is called what? Pre-proinsulin. Pre-proinsulin. First of all product is pre-proinsulin. The first part, because it's composed of 4 chains, the first part is called signal peptide, or some people call it initial peptide, initial sequence. This first part, first signal peptide, is cleaved by a special enzyme inside the endoplasmic reticulum. This enzyme is here and cleaves the signal peptide. This part is removed, which is called what? Signal peptide, by the enzyme called endopeptidase, or specifically call it signal peptidase. What remained? 3 chains of the pre-proinsulin. We said pre-proinsulin is 4 chains. The first chain is removed, which is signal peptide, and the 3 remained ones is called proinsulin. This proinsulin, after the cleavage of the signal peptide, is directly folded, and also the disulfide linkages form it. It will be like that. This is called what? Proinsulin. This is the pre-proinsulin. Then we have proinsulin, which is 3 chains of the amino acids, 3 chains of the peptides, the A chain, the B chain, and the C peptide. This part is called A chain. Here is B chain, and this is called C peptide. It's not called C peptide because it's C-shaped. No, because it's called connecting peptide. C for connecting the A chain and the B chain. And between the A chain and the B chain, we have 2 disulfide bonds, and 1 disulfide bond is here inside the A chain. This is called proinsulin, but still we don't have the insulin molecule, right? This proinsulin is built up from the endoplasmic reticulum, packaged in special vesicles, and transported into the Golgi apparatus. Okay? This built up from here. This vesicle contains what? Proinsulin. Okay? This is the proinsulin, and this is the vesicles from the endoplasmic reticulum. This is transported to the Golgi apparatus. Here we have the Golgi apparatus. And this special vesicle from the endoplasmic reticulum fuses with the membrane of the Golgi apparatus, and the proinsulin is entered the lumen of the Golgi apparatus. So now we have what? We have proinsulin inside the Golgi apparatus. Inside the Golgi apparatus, there are many enzymes, and 2 of them are important for us. The first one is called prohormone convertases. Prohormone convertases are special enzymes that cleaves the proinsulin into insulin and C-peptide. And also another enzyme is also important for us, which is called carboxypeptidase. Here we have also carboxypeptidase. Okay. The carboxypeptidase acts here, removes the amino acids, and the prohormone convertases, especially type 1 and type 2, acts here. And what is remained? The insulin, which is A chain bonded to B chain disulfide linkages, and also the C peptide is remained. So now we have what? We have insulin, and also C peptide. C peptide, which is about 31 amino acids, And the insulin is 51 amino acids. 51. 30 for the B chain and 21 for the A chain. And C-peptide is about 31 amino acids. Okay? The insulin and the C-peptide now fully matured and functional. Then they should be stored. They are stored in special vesicles or called secretory granules. This insulin and C-peptide bud off from the membrane of the Golgi apparatus, which contains insulin and C-peptide. Okay? Here we have many secretory granules which contains insulin and C-peptide. And because the C-peptide and insulin are first cleaved from the proinsulin. Proinsulin cleaved, break down by enzymes to produce two fragments. One of them is called insulin and the other one is C-peptide. That's why the insulin and the C-peptide are in the same amount. If we have 100 molecules of insulin, also we have 100 molecules of C-peptide. If we have 1000 molecules of insulin, we have 1000 molecules of C-peptide. And this called what? Equimolar amount. Means they are in the same amount. But there is one problem. The half-life of this secretory granules is about 5 days in the beta cells. And in the secretory granules, there are many enzymes that may act on the insulin, and during this period of the 5 days or maybe less or more, the insulin is degraded, break it down, and when it's released, it cannot act on the insulin receptor and exert its action on the blood cells. So there should be a way to store the insulin in a way to protect it from the enzymatic degradation. In the secretory granules, there are many transporters and channels. One of them is calcium channels and also zinc transporters. Here we have zinc transporters, all called zinc transporter type 8. This is important in the pathogenesis of the diabetes mellitus type 1. Anyway, zinc transported from the outside to the inside of the granule, and also calcium transported from the outside to the inside of the granules. Okay? It's positive charged. This zinc, 2 molecules of the zinc and 1 molecule of calcium can bind with 6 molecules of what? Insulin. This is the insulin molecules. One, two, three, four, 5, and 6. Each calcium atom and 2 zinc atom combines with 6 molecules of insulin, and this is called crystals of insulin hexamer. Hexamer means 6 molecules of insulin are united to each other, and because this is in crystals, means it's insoluble. It cannot be dissolved inside the lumen of the secretory granules. It's protected from the enzymes. As we said, we have many enzymes in this area, especially in the membrane. We have prohormone convertases and also carboxypeptidases and other enzymes. So in the secretory granules, we have what? We have crystals of insulin hexamers, which is stabilized with the help of 2 zinc atoms and 1 calcium. Enzymes, for example, carboxypeptidases and also prohormone convertases, and also a small amount of proinsulin that's still not degraded by the enzymes in the Golgi apparatus. Here we have some of the proinsulin. That's why we have enzymes carboxypeptidases and the prohormone convertases to convert as possible as of the proinsulin into insulin and also C-peptide. We have also what? C-peptide. These granules also contains other molecules, for example, free zinc ions, maybe release it, gamma aminobutyric acid, the neurotransmitter. It's act on the as a paracrine in the glucagon cells, the alpha cells, to inhibit the secretion of the glucagon, and also GAD, glutamic acid decarboxylase, and GAD enzymes. We said in the humans we have about 1 to 3 million of islet of Langerhans, right? 1 to 3 million. And each islet, which is composed of about few hundred of cells, and 60 to 70% of these are beta cells. Inside each beta cell, we have 5 to 8,000 of secretory granules. It's about 5 to 8,000 of secretory granules. And each granule contains 200,000 molecules of insulin. 200,000 molecules of insulin. This huge amount of insulin is estimated to be in the range of 200 to 250 units in the pancreas, and this is enough for the supply of a normal adult for about 10 days. But when the insulin is needed, this huge amount is not released into the blood. If it's released, a very severe hypoglycemia may result. Only few of the insulin can be released from the granules, and these granules that are active and able to release its contents, which is mainly contain insulin and other products, I'll call it readily releasable granules, or called mature cells. The remaining part, which is most of the granules, are called immature granules. Okay, let me repeat it. The beta cell, we have inside the beta cell in the chromosome number 11, we have the insulin gene. Insulin gene is first transcribed into the mRNA. mRNA diffuses out to the cytosol to be translated by the ribosomes, and peptide chain will form it. This peptide chain, because the first amino acids are too hydrophobic, recruit the ribosome, the mRNA, into the membrane of the endoplasmic reticulum, and this peptide chain enters the endoplasmic reticulum. And after more and more amino acids added to the chain, peptide chain, which is composed of 4 chains, formed with about 110 amino acids, and this peptide chain is called what? Preproinsulin. This preproinsulin, the first chain which is removed, is called of signal peptide by the enzyme signal peptidase, and the proinsulin is remained, which is the folded peptide, and also the sulfide linkages are also formed. Then this proinsulin is built up from the endoplasmic reticulum and enters special vesicles. This special vesicles goes into the Golgi apparatus, and inside the Golgi apparatus there are other enzymes, for example, prohormone convertases and also carboxypeptidase type E, to fully maturate the insulin and convert the proinsulin into insulin and also C-peptide. Then this insulin and C-peptide are stored in the secretory granules with the help of the zinc ions and also calcium ions, which is stabilized in the form of insulin hexamers, meaning 6 insulin molecules are attached to zinc ions and also calcium. And also in the secretory granules we have C-peptide, glutamic acid decarboxylate, GABA, free zinc ions, and also proinsulin and other enzymes like carboxypeptidase E and also prohormone convertases. That's all about the insulin synthesis.